Let's review the drug class of systemic antibiotics. The representative agent is ketorolac. It's indicated for acute, severe post-surgical pain. It works via potent prostaglandin synthesis block. First, let's review opioid analgesics. They bind to central nervous system mu-opioid receptors. Next, corticoids like dexamethasone. They suppress inflammatory pathways.
AI-generated prescriptions are for informational purposes only. They do not replace professional medical advice. Always consult with a qualified healthcare provider before making any medical decisions.
Forensic odontology is a specialized branch of dentistry. It evaluates dental evidence in the interest of justice. Teeth can survive extreme environmental challenges.
In pediatric cases, we track tooth development and mineralization. The neonatal line is key, determining if an infant lived postnatally.
The Demirjian method evaluates 7 left permanent teeth. Mineralization is classified into 10 stages. These estimate age with a typical margin of error. For adults, estimation relies on regressive changes. The Gustafson method evaluates 6 dental criteria. Each is scored on a 0 to 4 scale. The sum is entered into a regression formula. This gives us a precise age estimate. It measures deposits moving from the apex. Lamendin measures periodontosa and transparency. A non-destructive alternative, here is the adult formula. This method is highly effective for adult remains, yielding a standard deviation of plus or minus 3.3 years. It can estimate age with high precision.
Inside the stable dentin core of a tooth, L-aspartic acid slowly converts into the D enantiomer over time. This ratio estimates age with high precision.
Teeth hold a time capsule of our atmosphere. Enamel traps carbon-14 when it forms. We match this to the bomb pulse curve. This pinpoint reveals your exact birth year. Line analysis is a vital forensic tool. We microscopically evaluate pre- and postnatal enamel. Its presence gives an immediate confirmation of life. This distinguishes live birth, stillbirth in infanticide cases. We use the Demirjian method for children. It grades 7 left mandibular teeth. The margin is within 1 year. It evaluates overall growth. The Gustafson method. It evaluates aging in adult populations. Using the formula, age equals 11.43 plus 4.56x, with a margin of 5 to 10 years. The Lamendin method uses single-rooted teeth to estimate adult age. We measure transparency and periodontosis. It's accurate to within 3.3 years, a fast and non-destructive
The mandibular canedex, or MCI, is used on postpubertal adults. It divides canine width by intercanine distance. It is 80% accurate. It determines biological sex.
The discriminant canine index limits its target population to postpubertal adults. It maintains an accuracy of over 72%, serving as a key tool for sex estimation. Complex networks govern odontogenesis. They drive both variation and tissue inflammation. We must also address clinical gender disparities. This will help unlock full professional potential.
Disruptions in specific genes cause dental agenesis. Hypodontia is the absence of fewer than 6 teeth. Oligodontia is missing 6 or more. Anodontia is the complete absence of all teeth. MSX1 on chromosome 4 controls critical dental signaling. Mutations disrupt the bud-to-cap transition, causing agenesis of incisors and premolars, resulting in severe oligodontia.
Located on chromosome 14, the PAX9 gene is essential. It regulates BMP4, but specific mutations cause molar agenesis. Located on chromosome 17, Axin2 is a critical inhibitor. It regulates tooth number and size. Mutations cause severe oligodontia, linked to colorectal cancer syndrome.
The Y chromosome directly influences dental development. It stimulates cell proliferation in the dental papilla, increasing both enamel and dentin growth. This explains why male teeth are generally larger.
XXX females possess an extra X chromosome. They exhibit a much thicker enamel layer. Their mandibular roots are significantly longer, showing localized gene effects. Females with Turner syndrome exhibit a significant reduction in tooth size. This is characterized by thin enamel layers, smaller dentin cores, and shortened roots, affirming the dosage-dependent nature of X-linked dental genes. The AML gene on X and Y chromosomes provides reliable sex identification. It regulates enamel growth, mapping distinct markers for forensics. AML X and Y have key variations. PCR allows for rapid sex identification. 2 bands indicate a male, while 1 indicates a female.
PCR amplification of the SR gene identifies male remains. In degraded remains, LCMSins, this detects sex-specific isoforms in ancient teeth.
Inside developing teeth, LNCRNAs are vital regulators. In the nucleus, they scaffold complexes to control genes. DLX6 to become dentin-producing odontoblasts.
MicroRNAs are tiny regulators of gene expression. By binding to mRNA, they repress translation, causing severe dental defects, including enamel-free teeth and blocked cell differentiation. Gene 5P acts as a major regulator. It targets and downregulates RET and ZEB2. Disruptions can impair EMT, causing dental anomalies. Variants in MIR6
Inside dental stem cells, epigenetic modifications like DNA methylation and histone acetation regulate gene expression, driving their differentiation. Interestingly, DPSCs exhibit a unique profile. They retain large prembryo, making them a valuable model for disease. They help us unlock fetal origin diseases.
PDLSCs exhibit selective hypomethylation. DPSCs are primed for dentin pulp regeneration.
Inflammatory markers show sex-specific differences. Females exhibit higher IL-1 beta and OPG. Males show elevated MMP8, driving aggressive collagen breakdown. In South Asia, gingivitis is slightly higher in females, but severe periodontitis is more prevalent in males. At age 5, it's 50% in males. Adult caries are higher in females.
Genomic, transcriptomic, and epigenetic biomarkers are expressed in human dentition. Disruptions trigger destructive inflammatory cascades, providing clinical recommendations.
Dentistry is transitioning, moving to a personalized, biology-grading molecular science with care. Dental pulp stem cells can regenerate damaged tissue, reducing the need for invasive procedures. Dental programs must address the specific hormonal vulnerabilities of women therapies during pregnancy. Organizations must implement fair and establish transparent pathways for advancement. AMELX is a critical enamel matrix gene. It is located on the short deletion helps in sex profiling. It governs enamel thickness and mineralization. AMELI is an enamel matrix gene. It is found on the short arm of the Y chromosome. This gene promotes enamel and dentin development.
The SRY gene on the Y chromosome drives male sex determination. Located at position YP.3, it is a male-specific marker with 100% sensitivity.
It is a long non-coding RNA. It is active in the coronal dental papilla. This biomarker is vital in embryonic stages. It drives differentiation in dental pulp stem cells. Let's analyze the biomarker OPG, an inflammatory cytokine found in saliva. It is highly elevated in female cohorts, regulating bone resorption alongside MMP8.
Matrix metalloproteinase levels are tracked in saliva. These enzymes are significantly elevated in male periodontal pockets. They catalyze the breakdown of collagen,